Thank you and good day to everybody. I am Silvia Stacchiotti, medical oncologist at the National Cancer Institute of Milan, Italy. Well, within the cancer department, I work with adult patients affected by soft tissue bone sarcoma and GIST. And I really want to thank the Indian Society for Medical Oncology, the scientific committee, and in particular Professor Bai Pai for inviting me to join this conference today and to talk together about aiming the target in rare, in particular ultra-rare soft tissue sarcomas. These are my disclosure. And I think it is important to recall that based on the last WHO classification, we know that the family of soft tissue, bone sarcoma, and GIST incorporates about 80 different malignant tumor types, each of them characterized by different biology, prognosis, natural history, and sensitivity to systemic agents. In addition, we know that the majority of them are ultra rare, in particular based on a recent position paper by the sarcoma community of experts under the umbrella of the Connective Tissue Oncology Society. It was defined the incidence threshold to consider a sarcoma and ultra rare sarcoma types, which was set at approximately 1 or less than 1 case per 1 million inhabitants per year. And based on this threshold and the recent refinement of the incidence of several sarcoma types by the French colleagues, we today know that about 20% of all soft tissue and bone sarcoma cases belongs to one ultra-rare sarcoma type, and in particular that ultra-rare soft tissue sarcoma types are 56 different entities. These make very easy to understand that to develop a new target compounds, a new target agents for each of these sarcoma type, ultra-rare sarcoma type, is a real challenge due to both rarity and heterogeneity. And this is due, first of all, to the fact that they're less attractive for industry, especially when a new compound is already approved for another indication. And this makes this translate into few clinical studies, limited funding, very limited number of compounds specifically approved against a specific target in ultra-rare sarcomas, and a spread use of off-label drugs. Today, we have only 4 compounds, target agents, formally approved by FDA for ultra-rare sarcomas. These are tazemetostat for epithelioid sarcoma, pexidartinib for tenosynovial giant cell tumor, temsirolimus recently approved for PEComa, and avapritinib for the H4-2V PDGF receptor alpha mutated GIST. These drugs are formally available in the US, but they are not available, for example, in Europe, where only avapritinib is formally labeled for GIST mutated patient. On this basis, it is easy to understand that the only chance that our patient have to access those compound is as off-label drug or as alternative within clinical studies. On the other side, rarity makes the number, the amount of data on preclinical, clinical, and epidemiological aspects very limited. We have on this basis a really large degree of uncertainty when we need to take a clinical decision, and there is a great inconsistency in patient care across countries, across sites all around the world. How we can try to improve this scenario? Of course, by referring patient to center of expertise or networking networks of experts and increase the networking across sites, across institutions and countries. We also can try to improve the quality of care by drafting guidelines and position paper on each single ultra-rare sarcoma entity. We need to confirm diagnosis and to help referring to expert pathologists cases since initial diagnosis. We need to collect data, share databases, and start registries. We need to improve a lot our interaction with the pharmaceutical company and the regulators to try to raise the challenge in develop new compounds for ultra-rare sarcoma entities. And we really need to strengthen our collaboration with patient advocacy groups, since patients are the first to explain to regulators which are the limitation in treating their own disease. And finally, I think that is very important as a community to be ready to provide clinical, preclinical, and epidemiological data when a new target compound is available and started is started to investigation for a better study design in each ultra-rare sarcoma type. In this regard, it is very important to try to understand in advance which is the strength of the preclinical rationale supporting the use of a given target agent against a given target in each sarcoma type. It is in fact important to remember that it is not enough to have a target. It is important to understand which is the addiction of our tumor type to the target, since the probability of a positive result of an anti-tumor effect decreases with the decrease of the relevance of that target, and the relevance of a given target varies a lot across ultra-rare sarcoma types. Now I, of course, cannot make too many examples due to time constraint, but I want to show you how it has been possible to develop new target agents in ultra-rare sarcoma types, following this process of knowing well the biology of the disease, identifying the correct target, the correct target agent, and trying to design the correct study, clinical study, to get at the end positive results. The first example, I think, is PEComa. PEComa is a disease in which we knew many years ago that there is the presence of an activation of the mTOR pathway. At the beginning was not clear which was the reason for mTOR pathway activation, but today we know that this is more often due to the presence of a mutation to TSC1 or TSC2, based on which mTOR is constitutionally phosphorylated and active. Based on this evidence, a few years ago, the mTOR inhibitor sirolimus was tried in a patient, and the response to this compound was published. After this report, the community started using mTOR inhibitors as off-label compound, since sirolimus was already approved drugs for other indication, and there was not a clear intention by the pharma company owning the compound to develop the drug in this tumor entity. All the clinical data available on the activity of sirolimus and other drugs available for treatment of sarcomas like doxorubicin and gemcitabine were retrospectively collected by the community of experts, showing that mTOR inhibition is a good option for this patient, even though off-label, while conventional chemotherapy was not active, is not active at all. And this was followed by a prospective clinical study that was started once a new compound, nab-sirolimus, belonging to the same family of mTOR inhibitor, was developed. This was a window of opportunity, and on this basis, a phase 2 studies focusing only on PEComa was started. In this phase 2 study, there was not a control arm, but based on retrospective data, was possible to have at least a kind of external comparison with the inactivity of cytotoxic chemotherapy. And within those studies, approximately 30 patients with advanced PEComa were treated with nab-sirolimus for advanced disease, showing that approximately 0.5% of them got a dimensional response based on RECIST with a very long disease control. In addition, it was possible to investigate the molecular profile of those patients, showing that the presence of TSC2 mutation corresponds to a higher chance of response to nab-sirolimus, even though a few cases of wild type or mutated to SC1 patient responded to the drug. Based on these results, the drug has been approved by FDA and is now available for treatment of advanced PEComa patients in the US. The process to try to get the approval of the compound also in other continents is ongoing, and we really hope that this drug will be formally available to treat our patients also in Europe. Another example in which the community has tried to better understand the disease and collect data that could be now used for the approval of new target agent is intimal sarcoma. Intimal sarcoma is a tumor type that originates from the large vessels and the heart, characterized by the presence of MDM2 amplification. This amplification is shared also with wild type, with well differentiated, dedifferentiated liposarcoma, but these are two completely different tumor types. The community could show that anthracycline can be active in this tumor, but the duration of the benefit is limited, while the activity of gemcitabine and pazopanib, that are the other compounds available for treatment of sarcoma, is really marginal. This is a response to doxorubicin, and the first MDM2 inhibitors has been already preliminarily investigated, milademetan, and the results have been presented by our Japanese colleague during the ESMO conference 2021, showing some activity. With this basket study, 11 MDM2 positive and P53 wild type advanced intimal sarcoma patients were treated, showing some activity in terms of dimensional response and the duration of response in responsive patient. it is not yet very clear which is going to be the future process, the future development plan for this class of agent in intimal sarcoma, but of course they represent an interesting perspective for patient. And I want to finish with epithelioid hemangiendothelioma, again an ultra-rare sarcoma type in which the community of experts has tried to work out better which is the best clinical approach and which are the biological and clinical characteristics of the disease. This is a tumor characterized by the presence of a translocation that involves BVTR1, CAMTA1 or YAPTAZ, translating into the activation of the EPO pathway. A recent position paper on the clinical management of the disease has been published. Also, a retrospective international effort has been made to show that the conventional cytotoxic agent available for treatment of sarcoma are completely inactive in the disease, while sirolimus can stabilize progressive cases in about 50% of patients. During the last ASCO conference, were presented the results of the MEK inhibitor trametinib, with some responses, but probably not so much exciting results. What is very fascinating now is to know that if the translocation is available to activate the EPO pathway, we have now new compounds able to target the TEAD protein that is activated in epithelioid hemangiendothelioma as effect of the translocation. This protein belongs to the EPO pathway and can be targeted by target agents by different pharmaceutical company that will be probably started to be investigated in this disease in the next few months, and these represent the new perspective for this group of patients. We hope that what has been reported in terms of inactivity of cytotoxic chemotherapy will support the approval of this compound if these compounds, if they can be proven to be active in the disease. We have several other examples in which interesting targets could translate in the approval of interesting new target agent in soft tissue sarcoma. But as said, we need to work together to work out the biologic profile of each single tumor type, and to design appropriate studies with a good patient selection if we want to succeed and, as I said, get new compounds, new target agents approved for our patient. And with that, I want to thank you for inviting me, and I'm completely available for questions, doubts, and to discuss this topic further together. Thank you very much again.
Thank you, Silvia. A great talk. I will call upon Dr. Biswas to take the questions.
Thank you, Silvia. Thank you, madam, for giving me this opportunity. Thank you, Dr. Silvia, for this beautiful discussion, including the insight and the recent development in the rather ultra-rare soft tissue sarcoma. So we do see these cases in our practice, but again, it's very rare. We do have certain challenges in management of and diagnosis of soft tissue sarcoma, more so this rare subtype. So I have few questions and queries on this, if you can give your insight on this. So do you have any kind of diagnostic panel to identify those rare cytogenetic abnormalities or any other potential target in this rare subgroup of sarcoma? Because we do face challenges, and most of the time we do not have those FISH probe or the NGS facility to diagnose this rare subtype.
So first of all, thank you again for having me with you today. It's always my pleasure to work together. So in terms of diagnosis, so pathologic diagnosis and a correct identification of each sarcoma type, we usually follow what is indicated in the WHO classification. And so at our institution, we work out all the immunohistochemistry and molecular assessments that are considered the gold standard for each sarcoma type. So, for example, for epithelioid hemangiendothelioma, we always want to have the confirmation of the diagnosis by means of the positivity of the molecular assessment for the translocation. While in terms of search of the targets, these varies across histologies. So, for example, in a GIST patient, we are used to sequence the patient, the tumor sample, while we do not perform NGS or Archer or other deep molecular assessment for all sarcoma types. Of course, when there is the suspicious of entities like NTRK mutated sarcoma based on morphology, location, the lack of other clear market to confirm a more common diagnosis, we usually ask for example, the presence of NTRK translocation. But we are not allowed by our health system to perform on a regular basis a full, deep NGS or RNA assessment in our cases.
Yeah, thank you. So we do have same challenges. We don't have most of this advanced molecular testing or the NGS for each and every patient. So another aspect that you have discussed about this targeted treatment for this ultra rare subgroup, and there are few. You did mention there are few targeted agents approved by FDA in different ultra rare soft tissue sarcoma. Unfortunately, none of the drug is available in a country like India. I can recall that only temsirolimus is available in India if you have to treat PEComa. So access to care is one of the major challenges, and these are very rare sarcoma. So I believe there are some regulatory issue or the pharmaceutical issue that whether this particular drug will be launched in near future in India, we are not sure. So as of now, again, we are stuck to the treatment. We do treat them with sometimes with adriamycin, cytotoxic chemotherapy, and also pazopanib. So in a country like us or any other LMIC where those drugs are not available, where do you place adriamycin at all in treatment of this rare soft tissue sarcoma? And if role of pazopanib, we have seen few of the stability of the disease in some of the rare sarcomas like this vascular sarcomas, hemangioendothelioma, SFT kind of. So where do you place adriamycin or pazopanib when there is no standard of care available for these rare subtypes?
So, following also the ESMO guidelines for treatment of sarcoma, doxorubicin or anthracycline still remain our standard frontline. And I think that in the majority of soft tissue sarcomas, this is our first choice, both in the neoadjuvant, adjuvant, and advanced phase. Of course, there are histologic types like epithelioid hemangiendothelioma or alveolar soft part sarcoma, in which is very clear that this compound is really completely inactive. And on this basis, we really try not to provide a patient with a treatment that is toxic with no expected activity. From the second line, instead, the followed path varies a lot across histologic type and is really histology driven, of course, with the limitation of the compounds that are available. So, for example, pazopanib can be our first or second choice in histologic type like alveolar subparsarcomas or extraskeletal myxochondrosarcoma or solitary fibrous tumor, but this is not the case for epithelioid hemangioendothelioma, in which we know that anti-angiogenics do have really a marginal activity. So, I mean, based on the histologic type, we may select pazopanib or trabectedin or sirolimus or I mean also ifosfamide. It depends which is the expected activity of the drug. Then with regard to off-label compound, I think that we are not in a very different position compared to India, since our rules are pretty strict. So I mean, sirolimus is somehow cheap and can be afforded. But other drugs like, I don't know, crizotinib for IMT or, other target agents, MCSF inhibitor and so on, or also anti-angiogenics following pazopanib are not available. And this is really a discrimination for our patient. So we search for clinical studies in which they could receive these drugs. And on the other side, as a community, we try to provide all the data, hoping that a new compound will be available soon. A new pharmaceutical company will be interested in entering the market by means of our ultra-rare sarcoma type.
Thank you for sharing the experience and thoughts. So my last question, these are low-grade sarcoma with the indolent histology. Your comments on using low-dose metronomic oral chemotherapy for this patient when the patient cannot afford the TKI or some newer treatment?
This is probably not, metronomic cyclophosphamide is probably not one of the regimen that we more often consider, even, of course, in further line, this is an option. We probably, over the last few years, went back to reconsider gemcitabine, taxanes, the carbazine more than metronomic cyclophosphamide. But I think that, it is very important to share what we know in terms of activity of each of these compounds specifically in each sarcoma type. Once, we have the knowledge, even by means of retrospective information and common effort to collect retrospective data, it is at least easier to discuss with the patient what is available and then to take a common decision on what to do.
Thank you, Dr. Silvia, for this beautiful insight on this. So I don't have any more questions. So back to the chairperson or Dr. Jyoti for any further question on this. Thank you.
So thank you, Dr. Silvia. It was great. And when Dr. Silvia is there, the questions never end. But we have to, in the interest of time, move ahead. So I will hand over to Dr. Gundeti to conclude the session, and then we'll move to next.
Thank you very much for wonderful talk. I think really it's a very rare tumors with rare, rare possibilities, rare drugs we have. I think in the future, with as you mentioned, maybe next generation sequencing, target agent, definitely we will have the more targets and more drugs. Definitely we'll have the bright future. Thank you once again for giving this opportunity. Thank you, madam. Thank you once again.
Thank you very much.